Hello, everyone. My name is Veronica. I'm a graduate student at Sam Houston State University, and my project is based off of a research grant that I submitted a couple of years ago. And I was awarded the grant, and this is the project that I did with that grant money. And so my project title is The Characterization of Salmonella enterica serotypes, Heidelberg and Typhimurium Plasmids from human clinical isolates obtained in Texas. So I would first and foremost like to talk about antimicrobial resistance. So first and foremost, antimicrobial resistance is a very serious public health threat in settings such as nosocomial settings or in the hospitals, food production, and zoonotic transfer. And so in particular, nosocomial settings are practically, like I said, in hospitals. And so we're starting to see bacteria such as Staphylococcus aureus and Clostridium difficile, which thrive in hospital settings, start to exhibit antimicrobial resistance. We're also starting to see antimicrobial resistant bacteria emerge in food production. So as most of you can recall, like in the news, you hear of outbreaks of E. coli and salmonella in vegetables and meat. Well, this is practically one of these examples of food production and its role in antimicrobial resistance. We can also see antimicrobial resistant bacteria emerge from zoonotic transfer. So, from contact with animals to humans and vice versa. Every year, approximately 2 million people get antimicrobial resistant bacteria infections. And of those 2 million people, 23,000 of those people pass, unfortunately. And what makes things worse is these antimicrobials are not being produced by pharmaceutical companies because they see no profit in it. So in this image in the left-hand side, this basically talks about how we see the emergence in antimicrobial resistant bacteria. And so we're going to have a community and a population of mainly susceptible bacteria. And as a result of there being antibiotics, these resistant bacteria are going to thrive only because these antibiotics will not kill or inhibit the growth of these resistant bacteria, but it will for these susceptible bacteria. So we're going to see an emergence of resistant bacteria thriving in an environment. So next, I'm going to talk about horizontal gene transfer and the role it plays in the emergence of antimicrobial resistant bacteria. So we do see interspecies transfer occurring. There's this paper that was phenomenal in the sense that it actually found out that we do see bacteria such as Yersinia pestis and Yersinia rockerai transmitting genetic elements to bacteria such as Salmonella enterica, and that was phenomenal. And so we also see the transfer of antimicrobial resistant genes and plasmids along as some of those genetic elements. And so it could be picked up from the environment, it could be transmitted from other bacteria, and with the assistance of conjugal transfer genes and transposons, notably on plasmids. And so plasmid incompatibility, which is a phenomenon in prokaryotes, plays a significant role in how plasmids are transferred. And so basically plasmids with the same replicon typing won't be able to exist stably in a single cell line. But if there are different replicon types, they will be able to. And so the reason this is important is because these plasmids are able to carry these antimicrobial resistant genes and these transposons. And so this will allow for the emergence and spread of these antimicrobial resistant genes and plasmids. So next, I'd like to talk about Salmonella enterica. So, first and foremost, I'd like to point out my really cool picture on the top left-hand side. I actually took on a scanning electron microscope. It is Salmonella enterica. But yeah, it's pretty cool. So, first and foremost, yeah, Salmonella enterica is a foodborne illness. There are a wide range of symptoms, such as gastrointestinal distress to sepsis, which ultimately leads to total organ failure. And fortunately, the person passes because the bacteria is throughout the entirety of the human body. And the infection can actually be treated um, with usually nothing. Sometimes the person will just need to let the infection run its course, and other times in severe cases, antibiotics will need to be used. Approximately 1.2 million cases a year occur in the United States, and of those 1.2 million, 23,000 of those people will be hospitalized, and 450 people will unfortunately pass from these infections. And so the serotypes that I use in this experiment is Heidelberg and Typhimurium. And so these are two of the most common serotypes that we see in the United States. It's not saying that these are the only two, but there are plenty more serotypes. So practically, the objective of this study was to understand more about the properties of the multi-drug resistant plasmids and understand more about the properties in human clinical isolates. So we were able to obtain 147 total human clinical isolates from the Texas Department of Health and Human Services. And I was able to characterize the replicon type and determine the antimicrobials that the isolates were either resistant to or susceptible to. And so for my first hypotheses, it states that more than half the isolates with the replicon types will have AC phenotype. And so that is very common in human clinical isolates. For my second hypothesis, it is that over half of the isolates with the confirmed replicon types will exhibit some resistance to antimicrobials. And so for my methods, the first thing I did was I used replicon typing according to this really, really cool paper from Caratoli et al., where she actually developed a way to use multiplex PCR to detect these replicon types. And so I used that method that she used in that paper, and I also did an antimicrobial susceptibility assay. And so what I did is I just grew bacteria on a Mueller-Henson plate, and I just put antimicrobial discs around the plate, and I measured it according to according to the antimicrobial disc used, and afterwards, I measured it and used the CLSI guidelines to determine if the bacteria is either resistant or susceptible to an antimicrobial agent. And so yeah. So for my first result, this is for Salmonella enterica typhimurium, which is one of the serovirus that I initially looked at. And so as you can see, we do have some replicon types detected, and we also have some of these isolates exhibiting antimicrobial resistance. So of these 96 total typhimurium isolates we had, 31 had replicon types detected, and the most being FIA and FIB. FIA is actually seen in both food animals and human clinical isolates. And so of the 31 isolates, about 12 of these actually had resistance to antimicrobials. And so all of the antimicrobials, like ampicillin, streptomycin, chloramphenicol, sulfamethoxazole, and tetracycline, are all used to treat Salmonella enterica infections. And so it's kind of sad to see these resistance and alarming, so to speak. So next, these are from the Salmonella enterica Heidelberg isolates, and so this is the second serotype that I looked at. And so as you can see, there are replicon types detected, and almost all of them except for one isolate actually had antimicrobials detected. And so of the 50 isolates that we looked at, 23 of them actually had replicon types detected, and I1 and AC were actually detected mostly. Um, so I1 and AC are normally detected in human cases and also food animals. And so out of the 23 isolates, like I said, we only had one not exhibit any resistance to antimicrobials. And so all of these antimicrobials, streptomycin, tetracycline, and ampicillin, are all used to treat infections, salmonella enterica infections. And so majority of the samples did actually have replicon types detected, and sometimes some of these isolates did have more than one replicon type. And so this is normally seen in cases where an isolate is exhibiting multi-drug resistance. Um, with the isolates, with the confirmed replicon types, we did see some degree of antimicrobial resistance with some of the antimicrobials that were tested. So that was expected. And so for our first hypotheses, we're going to reject that. And our second hypothesis, we accepted that. And so for future reference, what we're going to do is we are going to observe antimicrobial resistant genes. We're going to screen for those, especially transposable elements and colistin resistant genes, which are newly emerging and problematic because colistin is a last resort antibiotic. We're also going to do conjugative experiments with Escherichia coli diatribe alpha. This will allow us to see if the plasmids are actually transferable. And we're going to screen for virulence genes, which we know that multi-drug resistant plasmids actually do carry. And these are my references. If anyone has any questions, please don't hesitate to ask. I hope you enjoy my presentation.